வணக்கம் மக்களே, let's get started. Today we are going to discuss about mediators of inflammation. Mediators of inflammation from chapter 3, inflammation and repair. Okay, what is mediators of inflammation? Inflammatory mediators are substances that initiate and regulate inflammatory reaction. Initiate and regulates inflammatory reaction. It is what given in Robbins. Okay, what are the most important mediators? Vasoactive amines. Second one is lipid products like prostaglandin and leukotrienes. Then third one is products of complementary system. Complement system like C5A. Okay, then fourth one is cytokines. Okay, these are the most important mediators. And how are mediators can be divided or based on the synthesis? There are two types: cell derived mediators and synthesized de novo. What is cell derived mediators? They are already preformed and stored in cells, in granules. They are stored in intracellular granules. So whenever they get any stimuli, it will get secreted into tissues by exocytosis of granules. Okay, this is cell derived mediators. And what are de novo, synthesized de novo? They are only synthesized in response to stimuli. Whenever they get any stimuli, they will start synthesizing these. Example: prostaglandin, then leukotrienes and cytokines. These are example of synthesized de novo. Then what are the major cell types that produce mediators? What are the major cell types? Whatever cells which can detect invaders and detect tissue damage, they can act as major cell types that produce mediators. What are they? They are macrophages, then dendritic cell, then mast cells. Okay, these are major type of cells. Then what are stimuli? We are talking about stimuli, but what are the stimuli? Stimuli may be anything including microbial products. It can be any microbial products, or it can be substance from necrotic cells. Okay, what are other things? One mediator can stimulate the release of other mediators. For example, complement system, products of complement system can stimulate histamine release. Okay, another example is TNF can stimulate the release of interleukin 1. So here these are secondary mediators. Here the secondary mediators can act same as like primary mediators, or they can act opposingly. They can even act opposing. Okay, so these are secondary mediators and these are primary mediators. It can stimulate the release of secondary mediators. Okay, next now we are going to deal with vasoactive amines. Why they are named so? What do you mean by vasoactive amines? They are named so because they are acting on blood vessels. Okay, what are the types of vasoactive amines? There are two things under this amines. They are histamine and serotonin. Okay, so what they are stored as? Histamine are stored as preformed molecules. They are already synthesized. They are already formed. They won't form after they get stimulus. They only release after they get stimulus. Okay, so they are stored as preformed molecules. Okay, then how they are So what are their source? Their histamine source is mast cells. Okay, so whenever they get any stimuli, it will get the mast cell will undergo degranulation and result in release of histamine into tissue. Okay, then what are the stimuli? Here the stimuli can be anything like a physical injury, then Ig antibodies, any antigens or antibodies, and it can be anaphylatoxins like C3A, C5A, anything. Okay, these can act as stimuli. And what histamine causes? What histamine causes? Dilation of arterioles, and another one is increase the vascular permeability of venules. How they do this? They do this by increasing the interendothelial gap. They increases the interendothelial gap through which the content will get out of the lumen. Okay, therefore it increases the permeability. It results in edema. Then for treatment we give histamine receptor antagonist. Here we deal with an integration, okay, horizontal integration with pharmacology. There are two types of histamine receptors. There are actually four types. Major thing is only two types: H1 receptor blocker and H2 receptor blocker. H1 receptor blockers are deal with allergic reaction, and H2 receptor blocker are given to reduce the gastric acid release. It is mainly given in peptic ulcer disease. Okay, so for allergic reaction we give H1 receptor blocker. In H1 receptor antagonist there are three generations: first generation, second generation, and third generation. We all know cetirizine is one of a it's a histamine receptor antagonist. Okay. Arachidonic acid metabolites. What are lipid mediators? There are two lipid mediators like prostaglandin and another one is leukotriene. These are lipid mediators. Okay. So what is arachidonic acid? Arachidonic acid is a 20 carbon polyunsaturated fatty acid. It presents in the phospholipid cell membranes, phospholipid bilayer. Okay, so whenever there is an inflammation, it results in the production of phospholipase. It is an enzyme which helps in the release of arachidonic acid from cell membrane phospholipids. So arachidonic acid will be get released, and there are two major classes of enzyme acts on arachidonic acid to form prostaglandin and leukotrienes. What are they? One is cyclooxygenase enzyme and another one is 5-lipoxygenase enzyme. Okay, so cyclooxygenase helps in the generation of prostaglandin, whereas the 5-lipoxygenase helped in the formation of leukotrienes and lipoxin. Okay, so the products like PGE, leukotrienes and lipoxins are called as eicosanoids which binds to the receptors to form binds to the G protein coupled receptors. Okay, both these things are collectively called as eicosanoids. So first we will discuss about prostaglandin. What is a prostaglandin? It is produced by prostaglandin is a lipid mediator, and it is produced by mast cells and endothelium cells and macrophages, etc. So there are two types of as I already said, cyclooxygenase results in the production of prostaglandin. So there are two types of cyclooxygenase like COX-1 and COX-2. What is the function of COX-1? COX-1 function is on GIT cytoprotective. Cytoprotective means it increases the mucus production and decreases HCl production, thereby produce the GIT cytoprotective. Therefore, it is cytoprotective. And renal cytoprotective by vasodilation, and it enhances platelet aggregation. Okay, then CO2, it's mainly confined to macrophages and which results in inflammatory mediators. So COX-2 is it is majorly confined to inflammatory responses. Okay, so there are in next speech. Okay, there are many products in this pathway like thromboxanase and prostaglandin PGI2, PGD2 and PGE2. How they are produced, each of which is synthesized by a specific enzyme acting on an intermediate in the pathway. So each of these produced due to specific enzymes. Okay, so the enzymes are restricted to have restricted tissue distribution. The enzymes has restricted tissue distribution. So what is the enzyme present in platelet? The enzyme present in platelet is called thromboxane synthase. Thromboxane sorry, spelling mistake, wait. Thromboxane synthase. Thromboxane synthase. So in vascular endothelium, it lacks thromboxane synthase and it has prostacyclin synthase. Okay, synthase. Thromboxane synthase result in the formation of thromboxane. It is a major product, TXE2, thromboxane. And prostacyclin synthase results in the formation of prostacyclin, PGI2, prostacyclin. It's a type of prostacyclin. Okay, what is their function? So prostacyclin, PGI2, it helps in it causes vasodilation and it inhibits platelet aggregation, whereas thromboxanase TXA2 causes vasoconstriction and promotes platelet aggregation. So they are very opposite to each other. They are totally opposite. It causes vasodilation and inhibit platelet aggregation, whereas it causes vasoconstriction and promotes platelet aggregation. Okay, that's it. Then so the thromboxane and the prostacyclin imbalance has been implicated as an early event in thrombus formation in coronary and cerebral blood vessels. Okay, this is very important. It is a Robbins line. Okay, so what is the function of prostaglandin D2? It increases vascular permeability and therefore it causes edema. And PGI2, it is a hyperalgesia, so it causes fever. Leukotrienes are produced in leukocytes and mast cells. Leukocytes and mast cells. And lipoxygenase is the enzyme which converts arachidonic acid into leukotrienes and lipoxygens. Okay, so lipoxygenase, there are three types of lipoxygenase. The predominant one is 5-lipoxygenase. 5 which converts arachidonic acid into 5-hydroxy-eicosA tetraenoic acid, 5-HETE. Okay, 5-HETE The function of 5-HT is chemotactic for neutrophils. Act as chemotactic for neutrophil. Precursors for leukotriene and lipoxins. Leukotrienes and lipoxins. So it is a type of LTB4 is a type of leukotriene. It is a potent chemotactic agent and it is an activator of neutrophils, and it helps in aggregation and adhesion of cells to endothelium. It generates ROS and release of lysosomal enzyme. Whereas other leukotrienes like LT, C4, D4 and E4 helps in vasoconstriction, bronchospasm, and increased permeability. So vascular permeability, leukotrienes are more potent than histamine to cause vascular permeability. Lipoxin, then we are going to discuss about lipoxin. The function of lipoxin is, it is very different to prostaglandin. Unlike prostaglandin and leukotrienes, the lipoxin suppresses inflammation by inhibiting neutrophil chemotaxis and adhesion to, it also inhibit adhesion to endothelium. These are the functions of lipoxins. Next, synthesis. Neutrophil synthesis is the precursor of lipoxin. Okay, that is 5-HT, and it will transport it to platelet where lipoxin is produced. Okay, thank you.